Hello everybody and welcome back to Microbiology. Today we will be covering bacterial culturing and identification lab. In medical microbiology, gram-negative rods play a very important role. There are a large number of pathogenic bacterium that are gram-negative bacilli or rods, and we have many different tests that are used to narrow down the identification of these bacteria. If you were to imagine that you worked in a laboratory and you were given a sample from a patient and your job was to identify a microbe, how would you go about doing this? Well, naturally you would want to perform a series of tests in order to try and see what sort of chemical and metabolic and physiological properties exist with this organism. Very commonly, first, a microbiologist might do a gram stain. And a gram stain will very broadly categorize the bacterium as gram-positive or gram-negative. And will also tell you the shape and size of that bacterium as well. Next, you might do culturing. Culturing naturally takes a lot longer than a gram stain. A gram stain it takes about 5 minutes in good hands to get complete, whereas culturing requires the bacterium to grow in the medium. So this can take a little bit longer, typically overnight, and this can occur on selective or differential media or both. And this provides additional data on the identification of an unknown bacterium. Furthermore, additional biochemical, immunological, or genetic testing may follow that looks at the physiology and metabolism of this organism. Microbiologists can use what are called dichotomous keys, and these are flowcharts that have lists of tests that need to be done, and based upon the outcome of those tests, which tests should be followed up. And this allows microbiologists to strategically perform tests and narrow down their inquiries to just the essential tests. There are several different types of gram-negative bacilli that we need to be aware of. We have the pseudomonads, the enterics, the non-saccharolytics, all of which are gram-negative. And they all look very similar if plated on just a basic nutrient agar plate. The pseudomonads are all but one species aerobes, and so they all need oxygen. And they do not make endospores, and they use aerobic respiration to catabolize sugars. Non-saccharolytics are aerobes, but they cannot catabolize sugar. Remember Saccharomyces, sugar-eating fungus, that means sugars. So non-saccharolytics cannot catabolize sugars. They use aerobic respiration to catabolize proteins instead. And some of them are capable of using a compound called sodium citrate and ammonium phosphate when proteins are not available. The enterics are a very common group of bacterium. They are facultative anaerobes. If you remember, that means they can do aerobic respiration or anaerobic respiration. They can do fermentation, and the fermentation will yield different wastes depending on the species. They are found in the gastrointestinal tracts of humans and animals. And some enterics are coliforms, which ferment lactose and produce lots of acid and CO2. An example of a coliform is Escherichia coli. However, not all enterics are coliforms. For example, Salmonella is an enteric but not a coliform. And there are many tests that exist to distinguish between enterics and coliforms. This is important because there are so many capable of causing disease in humans. So let's start looking at some of these tests. The first of these tests is the MRVP broths. This utilizes two tests using just one media. These broths are very rich in glucose, and they are used to distinguish enteric and coliform bacteria based upon the waste products of their catabolism of glucose. The possible products for these tests are mixed acids or acetoin. The first half is the methyl red test. Methyl red is a pH indicator that's often used in growing bacteria in different mediums. This particular example, the methyl red pH indicator, is added after incubation, and if the broth stays red, then the bacterium ferments glucose and converts the pyruvate from that process to mixed acids. For example, lactic acid, propionic acid, or acetic acid. And here is an example of those results. The tube on the left shows a positive MR result, meaning glucose was fermented and acids were produced. The second part of the MRVP test is the Voges-Proskauer test. And some enterics or coliforms can ferment glucose and convert the pyruvate to a compound called 2,3-butane-diol and acetoin as their waste products. The VP reagents are added after incubation, and they react with acetoin and produce a dull red wine color. So as you can see here, the tube on the left with that dull reddish wine color is an example of a VP positive test for acetoin. Okay, next we have Simmons Citrate media. This media has no organic nutrients. That means it doesn't have sugars, fats, proteins, or their building blocks. It does, however, have a sodium citrate salt as a carbon source, and this is considered inorganic. I believe it has to do with how oxidized that carbon is, but if you want further details on what makes a carbon source organic, you might have to speak to a chemist. It also has two other compounds in it. It has ammonium and it has phosphate as a salt, and the ammonium provides the nitrogen and the phosphate provides the phosphorus source, and these are also inorganic. Simmons citrate media tests the ability to grow aerobically using these inorganic salts, which is very unusual and would point towards a very small number of bacterium. Catabolism of these salts results in basic waste products, so that is waste products that are high in pH. And these waste products turn a pH indicator from green to blue. However, even just having good bacterial growth in this culture, even though we might not get a strong color change, just having bacterium that can survive on these inorganic mediums is considered a positive result as well. So here is a citrate test example. It has one positive result and two uninoculated controls. Next are the hydrogen sulfide indole tests. These are used to differentiate members of the Enterobacteriaceae family, Salmonella and Shigella. This tests for the ability to reduce thiosulfate into hydrogen sulfide. The product, hydrogen sulfide, can then react with the iron present in the medium, and this creates a black compound called ferric sulfide. Some bacteria can also metabolize tryptophan, and the byproducts of which include indole. A color indicator turns red in the presence of indole. Neither Salmonella nor Shigella can metabolize tryptophan, and only Salmonella can produce hydrogen sulfide. Therefore, if the organism was Salmonella, we would expect hydrogen sulfide to be produced, and as a byproduct, that black compound ferric sulfide, and a negative result for the indole tests. This brings us to the Enteropluri test. This is a tube that allows scientists to rapidly test a variety of tests in one single inoculation. So this tube has a variety of different mediums stored along this tube, and one can inoculate with a needle each one of these mediums and examine the results in order to determine what bacterium it might be growing. This is good again for enterics only. And oops, let me go back here. And it's done in this way. We have two caps. We have a blue cap and a white cap on either end. On the blue end, there is a needle that protrudes out and it has a hairpin loop around there. That is the handle end that you can grip with your fingers. And on the other end, we have a needle that is used to pick a colony off of a plate. So you unscrew both caps and you hold the tube kind of like a pen. And with this end, you would rub across a colony on a plate. Then with this other hairpin loop end, you would grasp it with your fingers and you would slowly pull this entire needle out of the tube. So as it passes across each one of these mediums, it will inoculate it with a little bit of bacterium sample. After you have done this, you push the needle back in most of the way, and there's a little notch on the left-hand side, which allows you to easily break off this end of the needle. And you take that end of the needle and you poke little holes in the windows of most of these wells, and that allows for air. You poke holes through little windows available on each one of these media tubes, and that allows air to get in so that aerobic respiration can occur. Except for, I believe, the last 4 windows are all fermentative, and so no oxygen is allowed into these tubes. So here we have a list of the different tests that this Enteropluri test tube will conduct. There are 12 different chambers and about 14 different tests that it will look at. The first one we can see is glucose and gas. So this tests the fermentation of glucose. If the fermentation of glucose occurs, then you will get a red to yellow color change. And if in the process it produces gas, you will have a gas bubble present. The next two, 2 and 3, look at amino acid catabolism. Specifically looks at lysine and ornithine, and will change from a yellow color to a purple color. Next is that hydrogen sulfide indole test. Another special carbohydrate called adonitol for the chamber number 5. Next is lactose fermentation. Lactose is a type of sugar, and arabinose is another type of sugar. Finally, sorbitol is another type of carbohydrate. Then we have our Voges-Proskauer test, and then something called dulcitol/PA. This looks at fermentation of the dulcitol carbohydrate and the breakdown of the amino acid phenylalanine. And then we have urea catabolism and citrate catabolism. And this is an image of the color coding for the entero tube, the pluri tube that we use. What you'll see at the first tube at the top is a tube that has negative results. So these are the colors of the tube before you inoculate with anything. The second tube underneath that one is if every result came back positive. So here we can see the color changes as well as potentially any gas bubbles that might form. Then finally at the bottom, we can fill out our key depending on the results of each answer. If there is a result, we will insert a number in this empty box here. Then we will add up all of the numbers and write the sum in each one of these circles. And we can use this code to identify which organism we have. I'll show you how that looks in the next slide. And here we are. I know this image is a little hard to see, so some of this you'll have to take my word for it. But we'll start from left to right, from the first chamber to the 12th chamber. In the first chamber, we have a yellow color. And so it says if we have a yellow color, we're going to write down the number 4 in the box below. So here we have a 4, and we do not have a gas bubble. You'll just have to take my word on that, but we'll put a 0 there. For lysine, we see that it's not a blue color. Remember, we should have a blue if it catabolizes lysine, and it remained yellow, so we'll write a 0 there. And for ornithine, we also have a 0. For H2S, we don't have that black production or indole. We also have a 0 for that. We don't have the red color that's formed either. So we leave all of those as a 0 as well. And then as we see, we continue filling out this chart. And now that we have all of our rectangular boxes filled, where we see these brackets and the circle is where we'll take the sum. So the sum of 4 plus 0 is 4. And then we have 0, 0, 0, and then 4 plus 2 plus 1 is 7. Okay, finally what you're going to do is you're going to take this code over to the Enterotube code book, and you'll do a Control F to do a find. Type in your code into the little find box and hit enter, and you're going to find what code links to what microorganisms. Many of them will have a single code for a single bacterium, but sometimes it doesn't provide any better narrowing down than 2 or 3 different microorganisms. If this happens, for your answer, you will just provide the code number you have and the 3 different microorganisms that are listed. Okay, so I am going to provide you with 5 unknowns. They're going to look like this. From left to right, you will see all of the different chambers. And your responsibility is to use the key provided here, the Enterotube key, and fill out the codes here and add up the little rectangular boxes to figure out your 5-digit code. Then look at the code book and discover what organism you have. On Canvas, you will have a 5-question quiz. For each question, I want you to write down your code and which microorganism or microorganisms it's specific for. And that will do it for today's lab. Thank you, everybody, and good luck. We'll see you next time.